Okay, now we're going to start chapter 3, and we're actually going to talk about some Mendelian genetics. I thought this comic was kind of cute. It shows genetics. This is how it works. Parent 1 with stripes this way, parent 2 with stripes that way. Baby gets a little bit of both. Okay, so you can't really talk about genetics without talking about the father of genetics, and that is Gregor Mendel. He did a lot of work with pea plants. So he was an Austrian monk. He studied science and mathematics at the University of Vienna, and he conducted a whole bunch of breeding experiments. The reason why he picked pea plants was because they were easy to breed. They had very distinctive visual traits or phenotypes, things like plant height, were they tall, were they short, pea color, were they green, were they yellow, texture, were they smooth, were they wrinkled, and so on. And so it's important to note that he formulated these fundamental laws of heredity in the early 1860s. And it's especially notable because he had no knowledge of a cell, what is a cell, or chromosomes. He didn't have a microscope. He just did all of this simply by observing the plants and what sorts of traits that they had. So some of the things that he looked at, and you can see some of his numbers here. First of all, seed shape. Were they round? Were they wrinkled peas? And when he crossed them, he found in the first case they were all round, and he counted them up and he did a bunch of math with it. He looked at seed color. Was it yellow? Was it green? The pod shape, the color, the flower color, stem height, and so on. And what he found in all of these was that the F1, we'll talk about what that means in a minute, but the F1 is just going to be the first cross that he did. They all showed the same exact phenotype or look to them. However, when he crossed them within themselves, he found a very closely standard F2 ratio of about 3:1 in pretty much every case, everything that he looked at. Okay, so the results that he had were very unappreciated during his lifetime. He was rediscovered in the turn of the century. We'll talk about some of his postulates. They're eventually accepted as the basis for Mendelian or transmission genetics. So before we go too much further, there are a few terms that we need to define. The first is phenotype. Phenotype literally means the form that is shown. It is the outward physical appearance of a particular trait. Now there are some things where you might not be able to see it, things like blood type, but it's the outward expression of a particular gene. Okay? The genotype is literally the sequence of DNA that's carried by all living organisms. It's the internally coded inheritable information. And so what this is going to refer to is the alleles that an individual receives at fertilization. So let's talk about alleles. Each trait is going to be controlled by at least 2 alleles, and alleles are just going to be alternate forms of a gene. These alleles occur on a homologous pair of chromosomes at a particular gene locus. So you might be looking at chromosome 1, a particular position there. You'll have 2 copies, one of which came from your mother and one of which came from your father. Now the version that you get, if they are both identical, it's known as homozygous. If those 2 versions that you got from your mom and from your dad, if they were different in any way, they're known as heterozygotes. Okay, so just to help you remember it, heterozygotes, just allele uneven. Okay, so his 4 postulates, and we'll go through these. First of all is that genes come in pairs, one from mom, one from dad. He didn't know that necessarily at the time, but he knew that the genes came in pairs and that they were inherited. The second thing is that these traits have dominance and recessiveness. So sometimes you can obscure or hide the expression of some of these in between different generations. Okay, we'll talk about segregation and independent assortment a little bit later when we start talking about multiple traits. So first of all, the first thing he did was he established these pure breeding lines for each of the traits, and we're going to call those the P or the parental lines. And anytime we do a cross, the very first individuals that we start with, we'll always call them the P generation. So what he found was that when you had 2 of the same line that were mated, the offspring were always identical to the parents. Okay, that was really important for forming these postulates. So next what he did was he selected a particular characteristic such as plant height, and then he mated 2 parents that were purebred for contrasting traits, so maybe a tall plant and a short plant together. And what he found was that 100% all of the offspring looked just like one of the parental lines. In this case, they were all tall. So offspring from this cross we call the F1 generation, the first filial generation. So the example we have here, okay, here's the P generation, here's a tall plant, here's a short plant. They're going to be true breeding lines. So what happens is when these plants go to form gametes, they have two versions of the chromosome, but it doesn't really matter because during meiosis, when it forms these gametes, the only thing that it can give is this capital T, in this case for the tall plant. In this case for the short plant, it's similar, but for opposite reasons, they're going to be giving the lowercase t. So when you combine these two gametes together and form this offspring, or the F1 generation, they are all going to look like this parent in particular, although they do have different gametes. And he only knew this, that they're hiding that trait, is that when he crossed these offspring, this F1 or first filial generation, what he found was that 3 out of the 4 of the resulting set of offspring, or the F2 generation, were the same phenotype as found in the previous generation. But the last quarter or so of the progeny looked like that original grandparent, that other parental phenotype in that original cross. So in this case, short. So when we're doing crosses between single traits in particular, we're going to use a Punnett square. Now, a Punnett square is just a bookkeeping tool, and what we're going to do is put all possible different kinds of sperm lined up on one axis, and all the different possible kinds of eggs are going to be lined up on the other axis. And so the goal behind this is to create every possible combination of alleles between the two parents so that it's placed within the squares. Okay, so in this case, we'll use uppercase P. Okay, so in this case, on the left would be the sperm. It has a capital P and a lowercase p. On the top, we'll put the egg, and it has a capital P and a lowercase p. And what you'll do is you will just literally follow, and you will fill in each one of these little squares. Okay? So at the end, what you would have is, in this case, 2 capital P's, a capital P and a lowercase p, capital P, lowercase p, and 2 lowercase p's. So just to show you a little bit more visually, when he looked at the pea plant colors, you can see here the purple, this would be the original parent, and a white flower. So this is lowercase p, these are capital P's. The only gametes, or only versions of the alleles that this purple plant could give was a capital P. Okay, so you could line those up here on the left. This plant, it has a choice of a lowercase p or a lowercase p. That's really not much of a choice. So you have to line those up here. The only possible thing that it can give to its offspring is this lowercase p. So filling in these charts, you'll have a capital P with a lowercase p, capital P with a lowercase p, and so on. And what you'll see is in every single case here, you're going to have a capital P and a lowercase p in this F1 generation. Now in this case, this purple happens to be something known as dominant, and we'll talk a little bit more about that in a minute. But a dominant is going to hide or obscure the expression of something that is going to be recessive. So what he did next, again, was he took this purple, he crossed them within themselves. So a purple from here, and he crossed them. Now, there's a little bit of a difference when you set up the Punnett square. So for this parent, it has a capital P and a lowercase p. Either one is equally likely to give to its offspring. So you have to write out both possibilities here, a capital P and a lowercase p. For this purple plant, it's the same thing. It has a choice of a capital P or a lowercase p. Now when we fill in the chart, we will start to see some differences. Okay, we've got a capital P, capital P, capital P, lowercase p, capital P, lowercase p, and lowercase p, lowercase p. This is the F2 generation, and it is again showing that 3-to-1 ratio of offspring that Mendel saw. Okay. Similar thing here, you could do the same sort of thing with, say, tall. We've got a capital T plant times capital T lowercase t crossed with the capital T lowercase t. If you go and you look at that offspring, again, you're going to see that 3-to-1 ratio of tall plants to short plants. Now, there are many cases where somebody may show this dominant phenotype, but we don't know what is their genotype. Okay, so if somebody has a recessive phenotype, they're always going to have to be homozygous because they need 2 copies of that particular allele in order to see that effect or that phenotype. However, if you have that dominant phenotype, it can be, if you don't know someone's genotype, you know, they might be a homozygous dominant, or they could be a heterozygous. It could be obscuring the expression of that recessive allele. And so really the only way to tell is to perform what's known as a test cross. So what you do is you're going to cross your unknown but dominant phenotype individual with someone who has a homozygous recessive genotype. Okay, and so there's 2 possible outcomes from this type of cross. And the only way that you can tell is to look at the offspring of this cross. So if you go back, okay, say you have this unknown one and 100% of the offspring are going to, in this case, be tall. What you can say then was that that original parent of unknown genotype must have been a homozygous for that trait. In contrast, if about half of the offspring were tall and about half of the offspring were short, you can say that that parent must have been heterozygous for that particular trait, because when you're looking at what potential versions of the trait that they can give, the heterozygous can give a tall and a short. Okay, this other homozygous, he can only give tall. So when you do this cross, you look at the offspring, this tall, no matter what, is going to obscure the expression of the dwarf trait or the short trait. Versus here, half of them will be tall and half of them will be that dwarf or short. Okay, so let's just practice a couple problems quickly. So in the first, go ahead and complete the Punnett square for the following cross.